Okay, thanks. So I'm going to present a method that my former student already developed, CellRBP, to predict protein-RNA binding in a cell-type specific manner. The work is still unpublished. So RNA binding proteins, right? They were the topic of the subject of several talks in the RNA meeting. We know that RBPs bind RNA, and they bind in a specific manner that depends both on the sequence and the structure of the RNA. And these RBPs play different roles in the cellular environment, including RNA transcript, translation. They affect stabilization, alternative splicing, as we heard in Chris Burge talk about splicing factors. And then the question that we ask and the researcher ask, where do these RBPs bind, where and when do they bind? And I think it's a question that also many of the audience here are dealing with, and this is what we want to solve. So indeed, experimental researcher have developed experimental techniques to measure where RBPs bind across the transcriptome. One of these experimental assays is called eClip. Now I know each people in the audience, some prefer eClip, some prefer ParClip, some prefer ClipSeq, but the reason why I present eClip today is that we're going to use it, and it's got the most comprehensive data set covering more than 100 RBPs in two different, or even more than two cell types. So that's a very attractive, having this comprehensive data set when we came to develop a new method. So eClip, for cross-linking immunoprecipitation, we get the binding for cross-linking of the protein to the RNA. Now through different processes, which I will not cover today, eventually we get reads that belong to the binding site. We can map them back to the genome, perform p-calling, and then a computational person like me goes to the database, to the encode, downloads a bed file. He can see and observe and extract the sequences that were bound. And here I'm showing an illustration of the sequences that are 8 nucleotides, but of course the sequences are maybe around 100 nucleotides. And also I'm highlighting a recurring motif which represents the binding site, but of course no one tells us where the binding site is or what is the binding site. So we need to find this recurring pattern. In many cases, computationally, we augment this data by negative examples, meaning unbound sequences, and then we define the binary classification problem that we need to solve to distinguish between bound and unbound sequences. But still, despite the fact that we have this eClip experiment, there are many limitations, right? Like any experiment, it takes time and resources, so we prefer a computational method. It requires an experiment for each RBP and each cell types, and there are many combinations of RBPs and cell types. We cannot do an experiment for each combination. And also, when you do the experiment, it doesn't cover all the potential binding sites. So for this aim, researchers have been developing computational methods to help predict where proteins bind RNA based on eClip data or based on other data. Here I'm referring to one such review, but there are many reviews, and there are many methods out there, and many methods developed by people here in the audience, as well as myself. But I will focus on only one method which we compare to. And the reason that I'm referring to this specific method, PrismNet, I don't know if the developers are here in the audience, because it uses cell type specific information. Many of the methods are based on sequence information or based on structure information, but usually the structure comes from a prediction by RNA fold, RNA fold, but not an experimentally measured structure. And here the unique approach that they took is that they used experimentally measured structure that you can get using an IC shape experiment, DMS, or whatever experiment you use. Here they show an architecture which they refer to as squeeze and extract a convolutional neural network with residual blocks, but I don't think the architecture matters that much. And eventually you get a binding probability, and you can also interpret the model and show where the binding site is, and I will go back to the interpretability later in the slides. So this is our competitor that we want to compete with. But still, what are the gaps? So it was a year ago that Mark presented in the RNA meeting in Lyon at ISMB his benchmark and review different methods. And one of the gaps that came up in his benchmark is a comparison between the prediction performance of different algorithms when they train and predict on the same cell type, and that's the x-axis, and when they train and predict on different cell types, and that's the y-axis. And you see that most of the dots fall below the diagonal, meaning it's much easier to predict for the same cell type, but not on another cell type. And this is a gap that we wanted to solve in this study. And also one of the ways that we are going to solve it is to use cell type specific information that many of the methods do not use, and I've shown one example of PrismNet. So this was the goal of our study, to develop a new method to allow cell type specific prediction using cell type specific information. Moving on to the data. So going back to eClip, we went to the ENCODE data set. You can download hundreds, or I think more than 200 eClip experiments from ENCODE that were published in this by Eric Van Nostrand, who was then a postdoc at Gene Yeo's lab. It's the most comprehensive data set. Again, we are solving a binary classification problem. There are 256 experiments, where the largest one included around 60,000 bound RNA sequences. We performed the same processing as was done in a PrismNet study. Namely, each binding site we trimmed or extended to 101 nucleotides, so that's the length of the sequences. In addition, we filtered out sequences where the IC shape measurements, which I will show soon define IC shape, the experimentally measured structure, were less than 40% of the sequence. We kept only the top 5,000 binding sites following this filtering, and then we added negative examples in a ratio of 1:2. So most of these data sets for each RBP and cell type would contain 5,000 positives and 10,000 negatives. And there were some cases that the number of positives was smaller, but then the ratio was still 1:2. Now moving on to other data that we used, IC shape data. So IC shape is an experiment that gives us in a single nucleotide resolution the structure of a nucleotide across the transcriptome. Now it's not exactly the structure, it outputs a score between 0 to 1 which correlates with the flexibility of the nucleotide. And flexibility is anti-correlated or negatively correlated with the base pairing. So if the nucleotide is flexible, it's likely to be unpaired. So this is an experimentally measured structure. We get it in a cell type specific manner. There was available data for both K562 cell types, both FG2, both cell types where the eClip experiments were performed. And unfortunately, in many cases, there are missing IC shape values for many nucleotides. So we performed an imputation using a simple neural network where we provided a sequence with the middle position that missed the score, the missing score, 41 nucleotides going through a multi-layered perceptron, and eventually we get a score for that middle nucleotide. These are the numbers of values we get for each cell type. We performed this in a training validation and test set that I show here, and the correlation that we got on the test set were greater than 0.5. So it got some prediction power, not the best, but still it helps us impute missing values in the IC shape dataset. Next data that we used was RNA abundance data. Here we are going for RNA-seq data that's in transcript resolution, so each transcript would have some value of its abundance. It was provided for different transcript isoforms. You can go to GEO or other repositories and download for both of these cell types. And one of the transformations that we had to do was a log transformation, because the FPKM values that show us the RNA abundance are highly skewed. And if you give these highly skewed values to the machine learning, it's not going to do well or use them in any manner. But once we do the log transformation, the distribution is much less skewed, and then the machine learning was able to use the log transformed values in an informative fashion. The last of the dataset or types of data that we used was RNA region type, and here I'm referring to different classes: the exon, coding sequences, the 5' and 3' UTR, which you can take from Ensemble. And then we used a majority annotation. Let's say we have a binding site with different classifications. We see that the exon has got the majority vote across the 101 nucleotides, and then we perform one-hot encoding to provide this type of information to our machine learning model. So now, to put this all together into one machine learning model, one neural network architecture. This is CellRBP, the model that my student developed, where we use two types of kernels, one short kernels and long kernels. The input is going to be the sequence information, which is as commonly done one-hot encoded. In addition, we added the IC shape score in one dimension, so this is appended to the one-hot matrix. We provide the RNA-seq, the log of the FPKM score. Following the pooling flattening, we have a concatenation layer that you see here, where we provide the FPKM, as well as the annotation of the region type. Eventually, this model, following a fully connected layer, hidden layer, outputs a binding score, but should be between 0 to 1 for the classification problem. There were 196 experiments that were used by PrismNet that we also used in this study. We performed hyperparameter search, which I won't go over the range today. We used the same partition of the data as was done in PrismNet, and turn time was quite fast, only 4 minutes. Okay, so now let's see how well we do when we predict across cell types using this cell type specific information. But before I go to the cross cell types, I do cross validation. Let's see how well we do on the same cell type. So we decided to leave out, and this is also commonly done, all the binding sites from chromosome 1 as a held out test set, and we used all the rest of the genome as the training. We evaluated this performance on the 196 experiments, and here are the results. So as you see, the average AUC of our model is almost 0.89 compared to PrismNet with 0.836. So at least we are the winners. We can put a V on the fact that we outperformed the competitor. But what's nice, we see that there is a lot of improvement by adding all of this cell type information that we were happy to see. And soon I will show you an ablation study where we added each type of information separately to show the effect. But now let's see if we outperform the competitor and how well do we do in cross cell type predictions. So we trained on one cell type, K562, and predicted on the other, HepG2, and vice versa. So that provided 124 experimental pairs of the same RBP, the same protein that was tested, and here are the results. So now the average AUC is 0.77 compared to 0.74 of PrismNet. So we are still we still have the gain, or we still outperform PrismNet. But what we can now see is the gap that still remains between cross validation and cross cell type prediction. So using this cell type information, we were able to close this gap in some manner, but still there is a gap and there's a lot of missing information. So this is what we are missing. So when we have this type of studies where, another point which is important, when we predict on one cell type, but instead of using the real cell type information, we provide the cell type information of the other cell type, we see the performance going down to 74. So providing that information gave us an average of 0.03 in AUC. So now going to the ablation study. In many cases where we provide different inputs to our model, we're interested in learning the contribution of each one of the feature types. So here we do it in cross-validation and across cell types. Let's first see in cross-validation. We start from a baseline of 0.846, which is for the sequence-only model. No extra features, cellular features. The biggest contribution was from the IC shape measurements, the experimentally measured structure, which brought us to 87. RNA abundance was the next most informative feature that brought us to 86, and the region type was the least significant feature. Still, it was nice to see that all of the features contributed significantly, and that the combination of all features got us to 0.89, meaning they provide orthogonal sources of information. Now going to cross cell type, are we going to see the same trend as we saw in cross-validation? So now we are starting from a baseline of a sequence-only model of an average of 71. We see that with the structure information, we get to 0.74. With RNA abundance, we get to 0.73. And then with the region type, we get to 0.72. So a very similar trend that we observed in cross-validation, we also observe in cross cell type. So that's comforting to see that we see the same order of contribution of the input features. And eventually the combination got us to 0.77 in average AUC. Now for the interpretation. We are not just interested in the black box and the prediction performance, but also to learn something about the biology. So we applied integrated gradients, which is a very established and common method in genomics to learn the important nucleotides. We applied it both on the sequence level with a baseline of 0, and both of the structure with a baseline that we used was the global IC-shaped median. We also converted each of the nucleotide, each of the structure symbols to P and U, depending of whether they were smaller or greater than this median. And we also performed global interpretation, looking at the preference for the region type, as also the preference on the sequence level in the form of integrative motif. So let's see how well we do in local interpretation. Local meaning we take a single binding site and we ask what are the important features in that binding site. So here is the interpretation for RB-Fox, and that I think the protein was also in Chris Bird's talk. You see that our model and both PrismNet picked up the binding site, but I think we got the initial U correctly, which PrismNet missed. And there's also some preference to unpaired nucleotides, which was also observed in PrismNet, but I think the structure preference is much less visible or clear than what we see in the sequence preference. Another example, it's HNRNPK. I think it's also one of the proteins we have seen in the talk before, the splicing factor. Again, you see that there is enrichment for Cs, both as identified by CellRBP and as identified by PrismNet, and it's less clear what is the structure preference in this case for the different Cs that are the binding site. Moving on to global interpretation. So we performed one interpretation looking at the preference for the region types, and we compared them to what was published in the Van Nostrand study in Nature 2020. And we see concordance in most cases. For example, AQR prefers to bind coding sequences. This protein DDX6 prefers to bind in the 3' UTR, and so on. So you can also observe the preference in terms of region type. And we also have a representation of integrative motif. So we took all the 7 MERS that had an activity score above some threshold, we aligned them, then we generated these motifs. You see a concordance here, for example, with the C-rich motif, and with RB-Fox2, these U-rich binders, HNRNPC and TR1. But here, with these cases, the motifs are kind of scrambled or not as clear, and there is less concordance between the methods and what was published in the ECLIP study. And this could be also artifacts or noisy experiments, and I think we also heard criticism about ECLIP experiments. So in the future, we plan to include RNA region type in single nucleotide resolution, and this was done in the DeepRipe method by Ganbari and Euler. Maybe add other additional information, such as evolutionary conservation and RNA modifications, which are known to be associated with RNA binding. We hope to be looking at better CLIP data, and there were some papers recently about improved post-processing of the data. One of the big challenges that we haven't able to solve, how can we use the in vitro binding models to improve in vivo binding prediction? Maybe we'll hear about in the next talk. And also the interpretation still holds some challenges, the structure preferences and global interpretation. So to summarize, we developed CellRBP that used these types of information that contributed in this order. Features were more influential than model architecture, which I haven't shown today. And now we can predict across cell types with some success, and that can reduce the number of experiments we'll need to do, and the method is publicly available. So with that, I thank you for the attention. The funding agencies, all is highlighted in the red rectangle, and I'll be happy to answer questions.
Thank you, Yaron. Any questions?
It's a very nice talk. I'll play a bit of devil's advocate, right? So you integrated some more signals to improve predictions. IC shape you mentioned about capturing structure, but actually the constraints are a combination in IC shape of the structure and whether it's available. And that has to do with RBP's binding. So I would argue that what you're getting is sort of second degree indications, whether there are bindings of RBPs in those specific locations, which would improve your predictions.
Right, so RBP footprint that we get from IC shape, right? And
yes.
Maybe on analog, that could be like open chromatin by ATAC-seq and what you get from ChIP-seq.
Yeah,
because
so you're going to see the sequence motif, and then you're going to get a footprint of whether the RBP was actually binding there, and you're going to say, hey, it's binding there. Well, okay.
Yeah, okay, so at least we succeeded in the prediction performance, right? Whether it was correlated or not.
Sure, but at a high price.
Causal,
but at a high price, right? Because now your model can only work with IC shape data, which is very rare, and you only get those readings for highly expressed regions. Then you need to impute, and that's very hard, as you mentioned.
Yeah.
So all I'm saying is, it seems a bit more complicated, and there is a price to pay at the same time. So is that okay. A fair comment?
But I think once you...
I'm not saying it's not good. I'm just saying
definitely.
that there is...
I'll give you the advantage. So definitely take my method with a grain of salt. But once you have IC shape measurements for specific cell types, now you can apply it to many RBPs. So that's the advantage, right? You do the experiment once, and the prediction for many RBPs in the same cell type. Yeah.
Very short one, then move to the next one.
Yeah, great talk. Maybe this is a naive question, but have you tested your method on guide RNAs used by Cas9 or other novel nucleases or base editors that have been identified, or are those sequences just too small? I guess that would give you, a data set other than the iSeq data that you're using to kind of test your method on.
Yes, so we use the ECLIP data, and in that respect, they only measured the binding of RBPs. I don't think they measured the binding of Cas9 as part of the ECLIP data set. There are other data sets that measure on-target efficiency and off-target, if that's what you're referring to.
Yeah, I guess using your model to predict where, whether or not, or where a Cas9 would bind a RNA, a guide RNA.
With a guide RNA, right? Whether it will. Okay, so I have
and
other.
That's cell specific, because that would be nice to know, because then you would know whether you would get cell specific.
Well, it's a big question that I have studies on, but that relates to CRISPR related studies, so we can discuss it later, I guess.
Yeah,
I'm happy to tell you.
Thanks a lot. And we'll move to the next.